DNA replication is a complex, multi-step process involving many proteins. The basic idea of it relies on the double-stranded structure of DNA and also on Watson and Crick's base pairing rules. Here is an overview of the structure of nucleotides. They consist of a phosphate group, a deoxyribose sugar, and a nitrogenous base. The two strands are antiparallel and held together with hydrogen bonds. Here is a simpler DNA model showing the base pairing rules. Each pair of nucleotides contains a small base, a pyrimidine, and a large base, a purine. Furthermore, adenine can only form hydrogen bonds with thymine and guanine only with cytosine. Thus, each strand can serve as a template for its complementary strand. Here is a representation of the replication complex of DNA. To start replication, helicase unwinds a section of DNA, called the origin of replication. It unzips the DNA, separating the strands, and primase adds an RNA primer to each strand. DNA polymerase III then adds DNA nucleotides to the new strand in the 5' to 3' direction. Here it gets interesting. DNA replication will occur in both directions on both strands, but remember that DNA is antiparallel. One strand must be synthesized in the 3' to 5' direction. DNA polymerase solves this problem by working in segments called Okazaki fragments. Each fragment must be primed separately by primase. Finally, DNA polymerase I replaces RNA primers with DNA nucleotides, and DNA ligase connects the Okazaki fragments. One final thing to consider: when helicase unwinds DNA, it builds up pressure past the replication fork in the rest of the DNA. This pressure would eventually prevent replication. To solve this, topoisomerase cuts DNA and unwinds it, then puts it back together. This relieves pressure on the DNA segment. Now let's talk about mistakes in the DNA, called mutations.
The first type of DNA mutation is substitution, where one base is switched out for another. So in this example, A is switched out for G.
An insertion is the addition of a nucleotide pair in a gene. This can cause a frameshift mutation, which occurs whenever the number of nucleotides inserted is not a multiple of 3. All nucleotides downstream of the insertion will be improperly grouped into codons. The result will be extensive missense, usually ending sooner or later in a nonsense and premature termination.
A deletion is similar to an insertion in that it can cause a frameshift mutation. However, it is different in that instead of adding nucleotides, it removes nucleotides.